There we go. All right. So today we're going to be talking specifically about how muscles work. So we're going to talk about microscopic structures. Just so you know, we're going to also be looking at our actin and our myosin and talking about how they actually form and how ATP is used within the muscles in order for us to actually move our body. Could someone please popcorn this out? Thank you, Andrea.
How do muscles physically work? How do muscles respond to stimulus?
Cool. So these are the three things that we will be talking about today. So, just a refresh. What do we already know about muscles and how do they function? Like, you go on to just like shout out a couple things.
Joint.
Muscles are attached to bones. Yeah, what else?
Movement.
They help with movement, right? What else? What else do we know about muscles? There's three types? Yep. Yeah, there's three types of muscle.
Skeletal muscle.
Cardiovascular.
Cardia. Yeah. And smooth. Yeah. Yeah. What else? Do you know anything about the structure of muscles or how it's organized? Yeah, there's different structures within muscles. You have that picture that has the blown up version of it. Right? Yeah, fascicles are a part of it. Our filaments are a part of it. Our muscle cells too. I just wanted to highlight two main connections.
Thick and thin layers.
Thick and thin filaments, yeah. Yeah. So two major connection points for our muscles. One, as Subin said, they connect to our bones via tendons. Okay, so this is this image. And then this one is what we'll talk about today as in neuromuscular junction. So it's where our neurons actually stimulate our muscles, which is why it's neuro, meaning neuron, and then muscular, meaning muscles.
Which one's this one?
This one?
Yeah.
This is just bone, like muscles attaching to bones. You could say via tendon, just to clarify, to make sure that you know that. The tendon is a specific type of connective tissue. It's this like kind of whitish, light pink part where the muscle actually intertwines with it and it's continuous.
So good.
Okay, fine. This muscles attaching the bone via tendon. Any questions or is it okay to move on? Yeah, I'll do silence and moving on. Alright. So, when we're talking about our neuromuscular junction, there's a couple different things. One, there are lots and lots and lots of different types of neurons. We're specifically talking about motor neurons. Because motor meaning movement, so the neurons that help us move our body, right? Because that's a huge function of our muscles. Now, these motor neurons actually help control the stimuli that are being sent from your brain to move the different parts of your body. Okay, and on the left, or sorry, on the right, I always get it mixed up. On the right, there's a picture of what a neuron, like a generalized neuron, looks like. Now, when we're talking about motor neurons, it's actually a very specific neurotransmitter, which is to say a chemical compound that our body creates, and it's called acetylcholine, otherwise known as ACH. So acetylcholine, when it's released, triggers the release of calcium 2+ for muscles to use during contraction. And we'll talk about why calcium is so important for muscles in just a second. And when we get into the nervous system, we'll talk more about the specifics of neurons and the different parts of them. But for now, just know that they're actually live cells that help send the signals. Any questions about motor neurons or acetylcholine? No? Let me know when you're ready to move on.
I'm ready for the number one.
Okay. I still see a couple people writing now. So this is just a picture from the textbook that I have. I just wanted to show you kind of what it looks like. So if you remember, this is like our muscle. Right, we have our actin, which is our blue, and then our myosin, which is like a purplish color. And then here we have our motor neuron. So our motor neuron actually connects via a synapse, which is the end of a neuron. And acetylcholine is released, which triggers a series of reactions that occurs within the muscle cells and muscle fibers. And all of these little things here are mitochondria. And muscle cells also need a lot of mitochondria because we're using them quite often, so we need that constant supply of ATP.
Wait, isn't there mitochondria in the heart as well?
Yep. Yeah, there's mitochondria in every single cell. It just matters what the concentration of that mitochondria is. Yeah, so our heart has the most mitochondria because it's consistently being used. Our muscles still have like a higher level, but it's not nearly as much as our cardio. All right. So talking about how muscles actually work, there's something called the sliding filament model, and this has been developed over many years, but pretty much it's saying that they filaments of actin and myosin, they slide past one another. So they don't actually change length, they just slide past one another. And the actin is the part that actually moves towards the center of the sarcomere. Remember, sarcomere is our functional unit of muscle. So sarcomere is from here to here. This is our sarcomere. Okay, so our actin actually moves in, and you can see that these don't change length, so it just gets closer together. So that's pretty much what the sliding filament model is demonstrating, is explaining. And then this movement, or sliding past one another, is also why there's a maximum for contraction of muscles. Like, you can only contract your muscles to a certain extent, and then you can't do it any farther. This is why. Questions about the sliding filament model? No? So what is the connection between neurons and muscles called? What did I talk about? Right, does anyone know what this is called? Yeah, Ezra.
It's called.
It's not our motor neuron. Our motor neuron is this part. But what's the connection between our neuron, our motor neuron, and our muscle? I'll give you a hint. The second word is junction.
Oh, well, okay.
Yeah. Neuromuscular junction, okay? So, neurons. Neuro, meaning a part of neurons. Muscles, meaning, like, our muscles. Muscular, muscles. So it's called the neuromuscular junction. Hey, there you go. Actually got it. I wasn't sure if I was actually gonna get it right. Okay. So, within our myosin and actin, there are actually specific structures and other like locations that are really important to talk about because they allow us to understand how the myosin and actin connect and how they actually will move. So our components are of myosin. We have our actin binding site. So here is where it actually will physically connect with the actin. We have an ATP binding site, ATP being our energy-rich molecule, right? We need that in order to complete contraction. And then it also has a head and a tail just for orientation purposes. So our myosin is pretty straightforward. It's our actin that's a little bit more complicated. And it's on the next slide. So our actin, there's actually 3 different structures within our actin filament. One, or I guess our binding site is, we have a myosin binding site. So on the head of the myosin, there's actually a part where that connects and it connects directly to our actin. And it's actually hidden in this picture because of the troponin. But our troponin is our, like that blue, the dark blue in the image, and it actually calcium 2+ binds there. So in order to complete contraction, you actually need calcium in your muscles. It has to be at a certain concentration that's actually high enough to support that movement. And then we have our tropomyosin, which covers the calcium binding sites until that concentration is high enough in the cell to support contraction. And then of course we have our actin filament. So just to clarify, we have our actin filament here, that's like in the middle. We have our troponin, which is covering the calcium 2+ site until the concentration is like up at a high enough level. And then we have our tropomyosin, which is that green braided molecule. Any questions about this? Or the components of either myosin or actin? No? We're going to go into the actual contraction cycle and see how this actually works. Alright, so generally speaking, when we're talking about contraction cycle, we'll start with relaxed muscles. Now, step 1 is when a nerve impulse is sent to your muscles, it triggers the release of acetylcholine in our neuromuscular junction. Right? So that's happening in that space and that connection point between our neuron and our muscles. Now, our acetylcholine causes our calcium to be released, and technically it's released from the sarcoplasmic reticulum, which is just a membrane that encapsulates the entire muscle. You just have to know that acetylcholine, once it's released, triggers the release of Ca2+. Okay. So this is our first step in contracting. You gonna go back? Or? Yeah. And again, this image is just showing you what it looks like microscopically. So okay, we'll move on. So our step 2. So our calcium concentrations in the muscle cells increases enough for our calcium to bind to the troponin, and then our tropomyosin actually will move out of the way and expose the binding sites for myosin. Okay, so our calcium connects to our troponin, which in return moves the tropomyosin, exposing the binding sites on actin for our myosin, and our myosin head actually binds to the actin. I also have a video that is later on in the slide so that y'all can actually visualize with this, because it's a lot of it's difficult to see without a video, especially since it's microscopic processes that happen. Alright, so okay, if I move on to step 3?
Of course.
Yes. Alright, so after our myosin heads attach to our actin, they actually pull on the thin filaments. Now, our ATP then binds to the myosin heads, which causes the myosin heads to release from the actin filament. So if you think about kind of like a tug of war, where you're trying to pull the rope. You got to imagine the actin is your rope, your hand is the myosin, and so you're trying to pull, and that's what happens. So the myosin will actually physically pull the actin closer. And there's a lot of, as you can see in this image, right, there's a lot of little bumps here. Those are the heads being visualized. And there's a lot of them that will continuously pull until that maximum contraction is reached.
Do they actually have heads?
Yeah. It's just that shape. It's not actually like a human head, but it's like a head. We just call it that.
Just to visualize it.
I'll look.
You know
I
my
was a
favorite
gummy
number.
worm or something.
Alright, so step 4.
That's the only number I pretty good.
So after our ATP binds to our myosin again, our ATP actually breaks apart into ADP and P, which releases our energy. So adenosine triphosphate is made up of adenosine diphosphate and another phosphate group. So that's why it breaks into that. And then as long as the calcium 2+ concentration and the ATP concentration are at a higher level, this cycle will continue. If not, then your muscles will relax and not continue through this cycle. Questions about these steps? On the next slide, I have another visual of what it looks like. I combined some of the steps so it was just 4 instead of 6, or if you really want to get into it, you could go even farther. Okay. Is it okay if I move to the next slide? So this is what our contraction cycle actually looks like. So here, we have our binding of myosin to our actin, and so it actually physically connects. There's something called a power stroke, which is just when myosin actually pulls the actin. Again, you have that now at a contracted location, like a contracted space. Our ATP binds, it releases, and then again it breaks apart that ATP. The energy there is now ready to connect again to the actin, and it continues going in that circular fashion. Video. Sit up. Any questions so far while I'm setting up the speaker? Does this make sense? What's up, John? Yeah.
Oh, cheap.
All right. Hey, the sound is really quiet. I don't know why.
Parallel overlapping fins, accidents, and myosin filament. The muscle contracts the new filament resulting in a short element structure, and thus the muscle is known as the sliding filament. Recycling forms the molecular basis for the sliding movement. Muscle contraction is initiated when muscle fibers are stimulated by a nerve impulse and calcium.
So this is our neuromuscular junction.
Covalent units on the actin myofilament are bound by calcium ions. The binding displaces tropomyosin along the myofilament, which is giant. Muscle contraction. Sliding movement. Muscle contraction.
So here. I'm just gonna turn off the sound because it's really bothering me. Alright, so our calcium binds to our troponin. And then it is able to move and expose these myosin binding sites. Okay, so that's what it actually does within your body. Now at this stage, it has the ADP and the phosphate group, which allows energy to be used to actually create a pull. And you can see once that ADP and the phosphate group leave, the myosin actually pulls the actin. And then new ATP molecules are then attached to the myosin. It makes the myosin release from that, and then it's ready to pull the actin again.
Wait, because you said move and expose.
So the calcium binds to the troponin, which moves the tropomyosin and exposes the myosin binding sites. So again, the cycle will continue to repeat.
And this is a good thing because it creates energy to pull.
I'll explain that in a second. Alright. So, our tropomyosin. So what does our tropomyosin do? Does anyone remember what it does?
It pulls and exposes myosin binding sites.
Yeah, so it covers our myosin binding sites. Right. Now, why would that be a good thing if we have our myosin binding sites covered part of the time?
Because it protects that?
Not fully. Think about would you want your muscles to be contracted the entire day?
No.
No,
Very
right?
uncomfortable.
So yeah, it'd be really uncomfortable. You'd probably be sore 24/7, right?
Also, it
So-
gives your body time to heal.
Not free from being contracted. So our tropomyosin covers our myosin binding sites. Okay. Now, when the calcium levels are high enough and the ATP levels are high enough, you're able to actually contract your muscles. But you don't want to do that all the time, which is why our tropomyosin, since it covers those myosin binding sites, is actually really beneficial for us. So that our myosin binding sites are not uncovered the entire day, so that we can actually have our muscles be relaxed and not contracting all the time and using up a lot of energy. Any questions about anything in the lecture? No?